Hi everyone, I'm Madam Tavasurya Ilangovan. In this session, I'm going to discuss about the second subtopic of the chapter 4, cell cycle and protein synthesis. The second subtopic of the chapter 4 is meiosis. Let's get started. Do you know what is meiosis? What are the importance of meiosis? And what are the stages of meiosis? These are the three main aspects that I'm going to discuss throughout this session. Meiosis is a process of cell division in which the nucleus of parent cell divides to produce four daughter cells. The four daughter cells that they produce during meiosis are non-identical to their parent cell. Parent cell is diploid cell, however, the daughter cells are haploid cells. Meiosis occurs in the reproductive cells, such as pollen and eggs in plants, sperm and ovum in animals. Human somatic cells are diploid cells. Diploid cell has two sets of chromosomes. It means total of 46 pairs of chromosomes. However, the human gametes such as sperm and ovum consist of 23 pairs of chromosomes. Out of these 23 pairs of chromosomes, 22 pairs of chromosomes are called as autosomes, and the remaining one chromosome is known as sex chromosome. Sex chromosome is a type of chromosome in which used to determine an individual's sex or gender, either females or males. Human females have 2 copies of XX chromosomes. However, the human males have 1 X and 1 Y chromosomes. There are 23 pairs of chromosomes. There is one chromosome is known as sex chromosomes. The remaining 22 pairs of chromosomes are autosomes. The autosomes are numbered by size. This is a structure of homologous chromosomes. Homologous chromosomes in which the two chromosomes composing a pair. The sister chromatids of these two chromosomes in a pair of homologous chromosomes known as non-sister chromatids. Importance of meiosis. Meiosis is important because it is a basis of sexual reproduction, and also it increases the variation in the characteristics of individual. The variation is due to the crossing over during meiosis. There are two stages in meiosis, such as meiosis 1 and meiosis 2. Cell divides by meiosis 1, produce 2 daughter cells, and then these 2 daughter cells divides by meiosis 2 to produce 4 daughter cells. This is the overview of meiosis. Meiosis have 2 stages, meiosis 1 and meiosis 2. However, both also have 4 main phases, such as prophase, metaphase, anaphase, and telophase. Since there are 2 stages, meiosis 1 consists of prophase 1, metaphase 1, anaphase 1, and telophase 1. However, meiosis 2 consists of prophase 2, metaphase 2, anaphase 2, and telophase 2. Okay, let me demonstrate the movement of chromosomes in meiosis. Before we watch the video, I have some questions for you. Let's challenge yourself by answering these questions after the video to recall back the lesson. Okay, let's rewatch the video regarding the movement of chromosomes in meiosis. The cell prepare before the divides by mitosis or meiosis. But in this video, I'm going to demonstrate about meiosis. So during the interphase stage, the cell have 2 centrosomes. Each of the centrosome have 2 centrioles. This is ester. Then the chromosomes starts to replicate and surrounded by nuclear envelope. This is the interphase stage before the cell divides by meiosis. Okay, after interphase stage, the cell prepared and can undergo meiosis. There are 4 main stages in meiosis: prophase, metaphase, anaphase, and telophase. Since meiosis have 2 stages of cell divisions, meiosis 1 have prophase 1, metaphase 1, anaphase 1, and telophase 1. In meiosis 2, prophase 2, metaphase 2, anaphase 2, and telophase 2. In this video, we focus the meiosis 1. Okay, after interphase, that is first stage of meiosis 1, prophase 1. Prophase 1 is the first stage in meiosis 1. During prophase 1, 2 centrosomes starts to move toward the opposite poles of the cell. While the 2 centrosomes move toward the opposite poles of the cell, spindle or microtubules extend out from the centrosome. Then the chromosomes begin to condense. Nuclear envelope disappear. After the chromosomes begin to condense, homologous chromosome pair up. Homologous chromosome pair up in a process called synapsis. After synapsis, the non-sister chromatids, it means sister chromatid from this chromosome and another sister chromatid from another chromosome in homologous chromosome, exchange their DNA segment. This process called as crossing over. So in crossing over, the non-sister chromatids in homologous chromosome exchange their DNA segment. The point at which the non-sister chromatids exchange their DNA is known as chiasmata. So chiasmata is the point where the non-sister chromatids exchange their DNA segment. Another pair of homologous chromosome also undergo the crossing over, prophase 1 followed by metaphase 1. During metaphase 1, the 2 centrosomes completely at the opposite poles of the cells, then all the homologous chromosomes line up at the metaphase plate. So the microtubule that attach to the kinetochore known as kinetochore microtubule, and some of the microtubules, one above and another one below the equator of the cell. Okay. So the microtubule that not attached to kinetochore known as non-kinetochore microtubule. So during the metaphase 1, keep in mind that all the homologous chromosomes line up at the metaphase plate. Middle of the cell is metaphase plate. After metaphase 1 is anaphase 1. Anaphase 1 is the third stage in meiosis 1. So during the anaphase 1, the homologous chromosome separate and move toward the opposite poles of the cell. This is anaphase 1. Homologous chromosome separate and no chiasmata. So after the homologous chromosome separate, the chromosome move toward the opposite pole of the cell. After anaphase 1, the last stage is telophase 1. During the telophase 1, the spindle or the microtubules depolymerize. The chromosomes completely at opposite poles of the cell. Then nuclear envelope reappear. This is telophase 1 in meiosis 1. Telophase 1 is the last stage in meiosis 1 and then followed by cytokinesis. Cytokinesis is a process in which the cytoplasm of parent cell divides by forming the cleavage furrow in animal cell. This is cleavage furrow in animal cell. However, in plant cell, there is no cleavage furrow. In plant cell, the cytoplasm of parent cell divides to form cell plate. Keep in mind that cytokinesis is not the stages of meiosis. Okay, after the cleavage furrow form, the cell completely separate to form as new daughter cells. The cell undergo meiosis 2 after meiosis 1. Therefore, there is no interphase stage. In meiosis 1, 2 daughter cells produce from one parent cell. Therefore, the 2 daughter cells continue meiosis 2. Even though 2 daughter cells, both also undergo these 4 stages of meiosis 2, such as prophase 2, metaphase 2, anaphase 2, and telophase 2. During prophase 2, each daughter cells have 2 centrosomes. Each of the centrosomes have 2 centrioles. This is ester. Then, nuclear envelope starts to disappear. Chromosomes become condensed and visible. 2 centrosomes will start to move toward the opposite poles of the cell. While the 2 centrosomes starts to move toward the opposite poles of the cell, spindle or microtubules extend out from the centrosome. After prophase 2, the second stage is meiosis 2 in second meiosis. Okay, during the metaphase 2, all the chromosomes line up at the metaphase plate or middle of the cell. Each daughter cells also undergo the second stage of the meiosis. This is metaphase plate or middle of the cell. During the metaphase 2, all the chromosomes line up along the metaphase plate. The microtubule attached to the kinetochore of the sister chromatid and known as kinetochore microtubule, and some of the microtubule that does not attach to the kinetochore of the sister chromatids, therefore it known as non-kinetochore microtubule. After metaphase 2, the third stage is anaphase 2. During the anaphase 2, the centromere that links two sister chromatids in chromosome breaks. Then the sister chromatids starts to move toward the opposite poles of the cell. Sister chromatids move toward the opposite poles of the cells due to the kinetochore microtubule. Kinetochore microtubule shorten and pull the sister chromatids toward the opposite poles of the cell. Anaphase 2 followed by telophase 2. Telophase 2 is the last stage of second meiosis, in which the sister chromatids completely at opposite poles of the cells. During the telophase 2 also, nuclear envelope form surrounding the sister chromatids. And then the spindle or microtubule depolymerize. Telophase 2 followed by cytokinesis. Keep in mind that cytokinesis is the process in which the cytoplasm of the parent cell divides by forming cleavage furrow in animal cell in order to produce 4 daughter cells. However, cytokinesis is not the stages of the meiosis. For a cell to complete the cell division, the cell should undergo the cytokinesis in order to separate as new daughter cells by forming the cell plate in plant cell, cleavage furrow in animal cell. Okay, this is the cleavage furrow. Okay. So during cytokinesis, cleavage furrow form and the daughter cells separate as new daughter cells. The daughter cells that they produce during cytokinesis are genetically non-identical to their parent. There are 4 daughter cells, and the daughter cells are haploid cells. However, the parent cell is diploid cell. Each of the daughter cell has only half as many chromosomes of the parent cell. Generally, meiosis 2 is equational division, in which the sister chromatids separate and form new daughter cells. Another one more, during the prophase 2 of the meiosis 2, there is no synapses, crossing over and no chiasmata. Compare then prophase 1 of the meiosis 1. The steps of the meiosis 2 also, most of the steps resemble the steps in mitosis. In meiosis, 4 daughter cells produced from 1 parent cell. However, in mitosis, 2 daughter cells only produced from the parent cell. The daughter cells they produce in meiosis 2 have genetic variations. The genetic variations among the daughter cells are due to the crossing over, random fertilization, independent assortment of the chromosomes.
Thank you and see you soon.